Hello everyone. Today we are going to learn about blood groups and physiological basis of blood transfusion. The human red cells contain numerous surface structures that are recognized as antigens by immune system. These surface structures are called as blood group antigens. Based on the presence of antigens on red cell membrane, corresponding antibodies are absent in the plasma of that individual. This forms the physiological basis of blood groups and principle of blood transfusion. The blood group antigens are called as agglutinogens. The antibodies against them are called as agglutinins or antibodies. As the reaction between them results in clumping or agglutination of red cells. There are more than 30 known blood group systems in our blood containing about 400 antigens. Many of them have cold antibodies that do not react at body temperature, therefore, only a few of them are immunologically active. ABO and RH blood group systems are not most important for are the most important for blood transfusion. The other major blood groups systems are MNS, Levi's, Duffy, Kell, P and Lutheran system. The red cells are less active cells that contain mainly hemoglobins. However, they play important role due to presence of antigens on their surface. Characterization of red cell antigen and antibodies form basis for compatibility of testing for blood transfusion. When the blood group of individual is known in emergency situation, blood can be transfused immediately with the suitable blood. Therefore, ABO and RH blood groups are usually displayed on the identity cards of individuals. The blood group antigens have provided the scientific basis for understanding hemolytic diseases of newborn and autoimmune hemolytic anemias. Blood group antigens play critical role in susceptibility to infections like malarial parasites, bacteria, and viruses. Red cell antigens are also associated with clinical disorders. Absence is associated with specific dysfunctions, inherited and acquired diseases. Medical legal experts take help of characterization of red cell antigen and antibodies in establishing uh, identify the father in the causes of in the cases of disputed paternities and mother in case of disputed maternity. Usually MNS will help. uh, Blood group is going to help. Specific blood groups have been reported to be associated with human behaviors as well. So they contribute to the personality of the individual. ABO blood group system. So this system was first blood group system that was to be known in 1901. The most important system. This is the most important system in transfusion medicine as ABO agglutinogens are highly antigenic. Physiologically, it is the most significant of all blood group system because of natural occurrence of A and B antibodies in the plasma of individuals who lack corresponding antigen on their red cells. In addition, the transfusion of incompatible ABO blood groups instantly leads to serious consequences. In ABO system, the essence is there are two antigens, antigen A and antigen B. Antigen has two subtypes, A and A1. The gene for antigens for ABO and ABO genes are located on the chromosome 9. These antigens are present on the membrane of red cells. They are also found in the tissues like salivary gland, testis, lungs, kidney, and pancreas. Therefore, they are, they are secreted in minute quantity in saliva, pancreatic secretion, and seminal fluid. Some individuals, especially blood group O, secrete blood group H antigens in saliva and other body fluids. So they are called as H secretors. The ABO antigens are present since fetal life. They first appear at 6 weeks of intrauterine life. At birth, the concentration is about one-fifth of the concentration in adults. The concentration rises slowly to reach a significant level at puberty and reaches maximum at 15 to 17 years of life. Therefore, the collection of blood for blood transfusion, the age of the donor should be minimum 15 years. Blood groups and the base based on their antigens. Based on the presence of or absence of A and B antigens on the red cell membrane, individually or in group, the four blood groups have been described. Group A antigen is present, Group B, antigen B is present, Group AB, both antigen A and B are present. In Group O, neither A nor B antigen is present. Okay, so there are subtypes as well. The A antigen has two subtypes, A and A1. The group A1 subdivided into again two groups. Group A1 containing A and A1 antigens and A2 containing A antigen alone. Similarly, the AB blood group is subdivided into A1B and A2B. In Indian population, the approximate distribution of ABO blood group is as follows: A blood group around 27%, B blood group 31%, AB blood group 8%, O blood group 34%. In A blood group, A1 is almost 75% and A2 is around 25%. Role of H antigen, H gene and H antigen. The expression of A and B antigens are dependent on the presence of H genes. Most of the individuals are homozygous for the H gene. The sequence of events in the formation of A and B red cell antigen is: A and B cell red antigens are glycoproteins and their formation starts with the basic precursor substances. The basic precursor substance is first converted to H substance by transferase under the influence of H gene. H substance is partially converted under the influence of A and B genes, their specific transferases, into A and B antigens respectively. Some of the H substance remains unconverted. Since O group individuals do not have A and B genes, neither A nor B antigen is found. Okay. And these have only H substance. A, B, and H antigens may be detected in saliva and other body fluids also. So such individuals are called as secretors, while the remaining they are called as non-secretors. So this is how A and B antigens are produced. So with the basic precursor substance, H substance is produced. Okay. So N-acetyl-galactosamine is going to attach to this H substance, which is going to form antigen A, and if D-galactose attaches to the H substance, it is going to form a B antigen. If neither A antigen or B antigen attaches to the H substance, okay, so which is divide of A or B blood group antigens, so it is going to form the blood group O, but it will have a H antigen. Bombay blood group, which has having a phenotype of O, discovered in 1952 by Bhende, Bhatia and Deshpande. So these individuals lack H gene and therefore basic precursor substance cannot be converted into H substance. Okay, so there is absence of H antigen. So this in turn results to failure of formation of A or a B antigen. Okay. So when their blood sample is tested for routine ABO grouping, they will be labeled as blood group O having. However, the serum contains anti-A and anti-B and anti-H bodies. So these individuals therefore should be transfused with only Bombay blood group. ABO system agglutinogens are of two types, anti-A and anti-B. The alpha agglutinin has two types, alpha proper and alpha 1. So these antibodies are found in plasma. Usually specific antibodies are formed against particular antigen when they are exposed to that antigen. But blood group antibodies are naturally occurring antibodies as they are formed without prior exposure to the antigens and are present in the blood of individuals in whom respective antigens are absent. Anti-A and anti-B agglutinins are not formed during fetal life and are absent at birth. They appear in the second week of neonatal life and increase very slowly in concentration to attain a peak level at about 10 years of life. It has been proposed that intestinal bacteria and food contain these antigens, similar to that of blood group antigens. Therefore, when the baby starts eating, food gets absorbed from intestine, the antigens are absorbed into the blood. So, as these antigens are recognized as a non-self antigens by the body's immune mechanism, they stimulate antibody production. So, however, these antigens are present in few concentration and antibodies against them are produced very slowly. So, therefore, antigen antibody reaction does not occur. So, moreover, antigens are removed by the phagocytic cells. The alpha and B agglutinins are globulins and belong to IgM category. Sorry. So, therefore, they do not cross placenta. They function efficiently at low temperature, that is between 5 to 20 degrees Celsius. Therefore, they are called as cold antibodies. Antibodies are directed against the antigens, as we know. The individuals with blood group A antigen on the red cell membrane do not have anti-A antibody, rather it possesses anti-B antibody in their plasma. The person with blood group B do not contain anti-B, rather have anti-A antibody in the plasma. Individuals with blood group AB do not have any antibody, and individuals with blood group O have both anti-A and anti-B antibodies in their plasma. So these facts were first noted and described by Karl Landsteiner in 1900, and his theory is popularly known as Landsteiner's law. This law states that if particular agglutinogen, that is antigen, is present on the red cell membrane of an individual, the corresponding agglutinin must be absent in the plasma. Conversely, if the agglutinogen uh, is absent in the red cells, the corresponding agglutinin antibody must be present in the plasma. This law holds good for ABO blood group system. However, the second part of the law does not apply for the RH and many other blood group as there are no naturally occurring agglutinins in this system. Thus, in ABO system, blood group A has antigen A on the red cell membrane and blood group B has antigen B on the red cell membrane and anti-A and anti-A antibody in the plasma. Blood group AB has both antigens AB on red cell membrane and no antibodies in the plasma, and blood group O has no antigens on the red cell membrane and possesses both the agglutinogens in the plasma. Yeah, okay, so as it is shown in the blood group A has a antigen and it has anti-B antibody, it has B antigen, it has anti-A antibody. Blood AB has both the antigens, there are no antibodies, whereas O has no antigens but it has both the antibodies. Blood grouping is performed by suspending red cells of the person's blood group to be checked in the anti-A and anti-B antisera that are commercially available. So, if the agglutinogen or the clumping as seen under the microscope is present, the red cells contain that particular antigen. Okay, so here it has shown how you can detect if agglutination occurs with anti-A, so that blood group is called as blood group A. If agglutination occurs with B anti-B, so then that blood group is called as blood group B. If the agglutination does not occur either neither with anti A or anti B, so that blood group is called as blood group O. If agglutination occurs with both anti A and anti B sera, okay, so then that blood group is called as AB blood group. The blood groups are genetically determined. Normally, the presence of blood group antigen is dominant characteristic. Therefore, the antigen is present in the phenotype irrespective of the genotype. There are three types of genes: O gene, A gene, and B gene. So, therefore, person can have any of the six possible genotypes: OO, OA, OB, AA, BB, or AB. So, gene O is genetically not important. Therefore, irrespective of phenotype, uh, maybe A or B or AB or O. For example, if blood group of one parent is A and other parent is B, so then there is a possibility of child having any of the four blood group of ABO system. Okay, so this is father with phenotype A, mother with B. Okay, so this is the genotype of father and this is the genotype of mother. So uh, the offspring can be of having any blood group. So it can be AB or A blood group or B blood group or O blood group. Okay, so these are the genotypes of blood group A, AA or AO, B, BB or BO. AB is always AB and O is always OO. In the next class, we'll discuss about the RH system.